So last, we will talk about how to perform the differential expression analysis for the single-cell RNA-seq data. So as we have mentioned before, so the single-cell RNA-seq may have many dropouts for each cell, which is randomly distributed for different cells. And if we plot the gene expression from two different cells, you can see that there are random dropouts like this has been labeled on these figures. And especially for these genes with a relatively lower expression level, it will be more likely to be dropout for different cells. And usually for the highly expression genes like some cell cycle genes or house-keeping genes, the dropout events will be less. So, but for the highly expressed genes, we will also see some over-dispersions between different cells. This is because that actually the transcription in each single cell can have some randomness, because that it might have some random initiation and durations, and also the risk of the RNA processing in each cell for each gene is different. So it might be slightly different for each single cell. And from this figure, you can see that actually the single cell RNA-seq data between different cells are very noisy, and it is relatively harder to perform the differential expression analysis. So how can we solve this problem? And especially solve the dropout problems. And so in the reality, we usually do not care about the differential expression between two single cells. We usually, because each single cell has so high dropout rates, we usually pull some cells together to compare the differential expression between different populations. And so, but even in different populations, you will still see some dropout events. And some people use the Huddle model, which is usually well applied in the economy areas. Let's assume that once you go to the supermarket, you want to buy something. So we can model it into two steps. One step is that whether you want to buy this thing or not. So this can be modeled by 0 or 1. So once you decided to buy these things, you can use another model to decide how many amounts you want to buy for these things. So this is similar for the gene expression calculation. So in this figure, so for these highly expressed genes, it's okay. It's just like you can compare the difference in the differential distributions. For here, we use a Gaussian linear model to model the gene expression and compare the difference between these two distributions to determine whether it is significant differential expressed. But for these genes with a relative higher dropout rate, we can separate them into two distributions. And we can first use a logistic regression to evaluate the number of zeros in each population for a single gene. For some population, it will have more zeros, and for another population, it will have less zeros. And then if we know that this gene is expressed, so for this part, we can use a Gaussian model to evaluate the difference between the different distributions. So actually, these two processes are independent to each other. So when you're comparing the gene expression, you can also perform some significance evaluation for the zero part, and also perform some significant evaluation for the non-zero part. Then you can combine these two significance evaluations because it is independent. You can combine them together to determine whether a gene is significantly differentially expressed between two populations. So that's the Huddle model mean. So, and in the single cell experiment, within each sample, we can calculate the differential expression. And we can calculate the differential expression between clusters. This might tell you some different marker genes between different clusters. And even we can calculate some differential expression within a gene expression cluster. Because some clusters, some very big clusters, they might be existing some subclusters. If you cluster them using a higher resolution, like previously you use k equals to 10, and now you use k equals to 20, you will get more clusters, and you will see some cluster with some subclusters, and you can also perform the differential expression between these subclusters. And we need to notice that one gene across the cells may have many zeros. And for the differential expression analysis, a very simple method is you use some tests, non-parametric tests like U-tests. And also we can use MUST, which is previously we introduced based on the hardware model to detect the differential gene expression. And also we can perform the differential expression across different samples. And when you compare the differential expression across different samples, a better way to do this is to average the gene expression of cells from same clusters. And then you can compare the differential expression between the clusters, between the average gene expression levels. And this time, because it is the average expression, it will have a less dropout. And you can even use some well-known methods in the bulk RNA-seq analysis like DSeq2 to perform the differential expression analysis. Okay. So last, we will talk about the single-cell RNA-seq applications. So, and the most common one is that we have already talked about is that you can use single-cell RNA-seq to separate different cell populations. So this is very easy to understood. And you can even find some real cell types, which is usually hidden by the bulk RNA-seq data. You cannot capture these real cell types by bulk RNA-seq. And also, you can use the single-cell RNA-seq to investigate some dynamic process, like you can investigate some drug treatment, some cells and some stimulations, and also investigate some cell differentiation process. So for this type of questions, the cells might be in a continuous status, not in the very distinct cell types. So you may observe some continuous distributions of the cells along the lines you are interested in. And so it will maybe reflect some cellular response to the stimulations. And finally, the single-cell RNA-seq can be combined with other technologies, because for most of the single-cell RNA-seq experiment, when you lysis these cells, actually is spatial information is lost in most cases. And so for some of the experiments, like for many of the questions related to the embryo development, you might combine these single-cell RNA-seq data to other imaging technologies for which you know the expression position of the genes, and you combine them with the single-cell gene expression data, so you will know the spatial distribution of this gene and also spatial distribution of these cells. Like this is the analysis performed in the zebrafish embryo, which is usually a ball. You can see the distribution of these cells. So this is very useful for the for some developmental studies, and also very useful for some immune-related studies and cancer-related studies to know the position of the immune cells and the cancer cells. And so finally, there are some useful pipelines for the single-cell RNA-seq analysis. So which most of them start from an expression matrix, and it like SURAT is the most well-known, and most people use that for the single-cell RNA-seq analysis. So it has many functions. It can help you filtering, starting from expression matrix. It can help you filtering the cells, perform the normalizations and the clusterings, and also the t-SNP validations, and also perform some differential expression analysis. And also another tool is named the Pagoda. The Pagoda has an online web face, which is an interactive web face. You can just use the Pagoda t-SNP, and you can select some cells from this t-SNP plot to do some more specific analysis if you are interested in some of these subpopulations. And at last, the link here, we provide almost hundreds of different single-cell algorithms. And if you are interested in, you can go to this website site to see the details. Because this is an area and very rapid development, and many people are developing algorithms for this. And for single-cell RNA-seq, tons of different methods are developed, sequencing methods are development. So let's summary for today's lecture. And so today we learned about single-cell RNA-seq technology and their analysis methods. So we know that the SMART-seq protocol have a better gene coverage, and but it can only perform one by one, so it can only perform on hundreds of cells. And we know that the droplet-based methods is more automatically and can perform on the tens of thousands of cells, but it has more dropout, both from the cell level and from the transcript level. And also, anytime if you want to analyze your single cell RNA-seq data, you need to remove the low quality and outlier cells first. So then after that, you can perform some downstream analysis and use PCA and t-SNE for visualization, and also use some canonical clustering methods to separate them into different clusters. And also annotate these clusters by the marker genes in the cells, or by compare the gene expression level of each cluster to the bulk RNA-seq expressions. And also, we need to mention that in the single cell RNA-seq experiment, it is very important to check the batch effect before you perform any downstream analysis, or your findings might be related to the batch, not to the real biological findings. And last, for the single cell differential expression analysis, you may need some specific models, like the Huddle models, to solve the dropout problems. So that's all. So any questions? Yeah. Mm-hmm. So the question is that for the UMIs, are the UMIs identical for the same transcript across different cells? Yeah, so actually the UMIs for the transcript across different cells is also different. So the UMIs is just one specific UMI is connected to one specific transcript. So when it's connected, if you perform some PCR analysis, then you know that the transcript is from the same UMI. But it is different between different transcripts. So then if the transcript is from the real expression level, from the real expression abundance, so it will have different UMIs to label them, and even for different cells. Yeah, because it is a combination of DNA barcode. Because for UMIs, it's also usually 8 base pairs or even longer. So it has many combinations. Yeah. So the question is that how UMIs and even how the barcodes is synthesized. So actually, you can synthesize it by random. So let's assume that if we have a base position that both started from A, so for the first base pair, it's both started from A. But you can put these four like DNAs in 4 lanes. And for each lanes, you can just, for this lane you add A, this lane you add G, this lane you add C, and this lane you add T. So you can get 4 different. Then for the next round, you can separate them into 4, also 4 directions. And for each round, you can just add a different nucleotide to this DNA barcode. And in the final steps, you will get millions of different combinations. So it is just synthesized by different rounds. Yeah, it is very to synthesize from the companies. Yeah.